Hi everyone, I'm Scion, and I'm currently studying chemistry at the Georgia Institute of Technology, and I'm also an assistant instructor for BIOS 1107, so Biological Principles 1, and this is a review of our first unit on life and evolution and how those processes occur. And I think that this is a really great overview of this unit, especially for any college class in biology or AP Bio, unless that course is at UGA, obviously. So, just for your reference for your studies, these slides were made off of the learning objectives found in our textbook online for free from the Biosciences department at Georgia Tech. And if you personally prefer a textbook that has a lot more in depth information and really writes things out in a narrative paragraph form, the Concepts of Biology textbook is great for that, and it's also available online for free. Okay, so starting off, our first section is on life, and it goes through what life is to how things evolve. And personally, I do wish that crocodiles evolved into crocs. They're a really great shoe. Okay, so moving on, we're immediately going to start with a practice question on what the requirements are for something to be considered alive. This is just one of those things that you have to memorize, and there are also kind of arguments about what the criteria are. So, five is more of the basic foundation rather than a set number of things. So, going through what I want to do is I want to go through our answer choices and just rule out things that are obviously false. So, looking at answer choice B, we see right here it says organization into tissues. That's obviously false. Things don't need to be organized into tissues because bacteria are alive. Do they have tissues? No. Let's again look at C. So, do we need energy? Yes. Do we need genetic information? Yes. Do we have to replicate? Yes. Do we perform cellular respiration? Well, that's not the only way to get energy, so we can go ahead and rule that out. And we can also rule out adaptation to cold climates because that's obviously not true. And then looking again at answer choice D, organization into membrane bound cells, yes. Evolve, yes. Respond to stimuli, yes. Need for energy, yes. Do we need a nucleus? No. Again, prokaryotes are bacteria and our archaea do not need a nucleus, so no, we don't need that. So lastly, looking at answer choice A, yes, we need energy. Yes, we're organized into membrane-bound cells. Yes, we store genetic information. Yes, we replicate. And yes, we change over time. So just again, commit those to memory because those are the five basic requirements for something to be considered alive. Okay, great. So moving on, now we can actually talk about what the characteristics for life are. Again, on this slide listed out are six. Just try and commit those to memory. So all living things have a need for energy. Okay, so that's the first thing. We need energy. We're organized into membrane-bound cells. We store genetic information. We can replicate, change over time, and these are combined growth in response to stimuli. Okay, so again, those characteristics, when viewed together, that's what makes something alive. And I can pretty much guarantee that on an exam, there's going to be a question about things and whether or not they would be considered alive or not. So then within life, one of the emergent properties is that of evolution. So what does it mean to evolve? And we need to really pay attention to this. It's the change in the allele frequency over time. So it's a change in the gene pool of a population, not in an individual, in a whole population. So again, changes in allele frequencies over time. That's like a very important concept to this entire unit, so just try and commit that to memory now. Before we continue on, I just want to go over some vocabulary. So if you hear the word genotype, what that means is the whole set of DNA of an organism. So every single piece of DNA in us as humans is our genotype. Now, when we talk about our phenotype, what that is the expression of our genotype. So that's when all of our DNA is made into proteins, and that's what we see on the outside as traits. And when we talk about genes, that's a specific segment of DNA that codes for a trait. So it codes for the physical expression of something. For example, hair color. Now, an allele is a variation of a specific gene. So, an allele, if we're talking about the gene for hair color, an allele is all the different variants for that. In this case, black versus blonde hair. So, the allele is the multiple different types or forms that a gene can take. Of course, we have the allele for hyperpigmentation versus normal skin here, and that's again just kind of demonstrating the different variations of a gene. And we just need to make sure to keep that in mind throughout this video where I say genotype or phenotype or gene or allele, and just try and keep them sorted out in your mind and know these definitions, especially for our genetics unit coming in the future. Okay, so again, we already understand the characteristics of life, and hopefully we can be able to distinguish that from something that's non-living. Here's just a greater list of all those things are. Again, it's a really thought out list. Just try to remember the 5 main characteristics. And then again, evolution is an emergent property of life. All things or all organisms evolve. And so what this means is that from mutations, natural selection, or other random processes, organisms will adapt and change over generations. So the big idea here is that evolution is change over time. That's where that characteristic comes from. And then we're talking about the 3 domains of life. So there are 3: bacteria, archaea, and eukarya. So bacteria and archaea are prokaryotes, which means that they are without a nucleus, and eukarya have membrane-bound organelles in a nucleus. So just be able to differentiate between those two right now. Prokaryotes are bacteria and archaea without a nucleus. Eukarya have membrane-bound organelles in a nucleus. Hopefully that should be pretty simple. Okay, so here we are at one of those questions where you have to distinguish if something is alive. Let's look at our answer choices. So the first thing we see is a robot. It can move, it can process energy, it can perform programmed tasks, but it cannot grow or reproduce. Therefore, is it alive? Obviously not. No, it is not alive because living things have to grow. Okay. Secondly, we're asked about a plant. It bends towards sunlight, so there's some form of movement, uses photosynthesis, so it generates energy, maintains internal water balance. What do we also know that as? We know that as homeostasis. Okay? It grows from a seed, and it produces seeds for reproduction, so as a way of reproducing. Okay? And so this fits all the criteria, so we know it's the plant, and then we can rule out the rest pretty easily. So we know it's B. We can rule out C because it's a virus. It has to use the host machinery, and it cannot. It obviously has no ability to produce energy. And then lastly, we're talking about a crystal. Hopefully, no one thought it was a crystal. Obviously not alive. There we go. Okay, so now evolution. So just like for life, evolution has a set of requirements for something to be considered alive. And so what it is evolution requires inherited characteristics and also requires variation among individuals. And this last part, the competition for limited resources, that's basically saying a selective pressure. So what that means is that a specific trait or a specific set of features are favored, and that means that things are going to want to change to be more like that. That's where we get inherited characteristics, and variation creates the means for that change to be possible. Okay, distinguishing evolution. Okay, yes. So biological evolution is a change in a population over a generation. Okay, whereas if something changes in a lifetime, it is not going to be inherited. So we have generations versus lifetimes, okay? This is a really key concept for what evolution is. Remember, evolution is a change in allele frequencies over time. So if we're looking at an individual's lifetime, is that evolution? No. For example, if you go to the gym every day and you work out and you get super buff, have you evolved? No, because that's just a change within your lifetime. And how do we know that things have evolved? Well, we have physical evidence in the form of geological and fossil records. If we look at things today, there are similarities that show how things have changed and remained the same over time, and we can sequence DNA now. And then the first piece of evidence for evolution was the Galapagos finches. Why anyone would want to study finches, I don't really know. I mean, Darwin did whatever he wanted to do, and now we have evidence for those. Okay, so again, we're asked which of these illustrates the requirements for biological evolution. So let's look at these, okay? So we have a population of bacteria. They're exposed to antibiotics. So what are those antibiotics? A selective pressure or a selective event. And what happens is that over generations, so not just in their life, through mutations and natural selection, they become resistant. Okay, so right now I'm looking at that. We have a change over time, over multiple generations. We have a change in the actual allele frequencies, so a change in the genes due to a selective pressure. So right now, to me, this looks like the correct answer. So, but just to go through and rule out the rest, you have a caterpillar becoming a butterfly, and obviously, when that happens, it's going to change physically and behaviorally, and it passes on those traits to its offspring, right? And then we have trees that grow taller over time due to the availability of sunlight. And this happens with individual trees stretching its branches towards the light. But again, individual trees, is that a genetic change? Is that on a genetic basis? No. And then, so it's obviously not C. And then we have a dog that performs tricks via a learned behavior. Is a learned behavior changing your genes? No. Is it passing that on? No. So it's not D. And then going back to B, looking at it again, a caterpillar transforming into a butterfly. Is there anything genetically changing during that? No. All caterpillars will, well, not all, but it's caterpillars become butterflies. That's commonly known. That's not a change in genetics. That's just a change during a lifetime. Okay. So the answer is A. Yep. Okay, so moving on to some defining features of evolution. So we're asked to differentiate between homologous and analogous structures. So basically, these are asking for a difference in ancestor. So when we're looking at homologous structures, these are going to be features that share a common ancestor. All right, so when we're looking at a trait of an organism, they come from a common ancestor. I know I just said that a bunch of times, but it's important to be able to contrast that to analogous structures, which evolved independently, which means that there is no common ancestor. No common ancestor. So, for example, an analogous structure are wings in birds and wings in bats. Do they have the same function? Yeah, pretty much. But did they evolve at the same time? No, they evolved independently. Whereas homologous structures, where we'll look at a tree of traits, we'll see that those evolved together in the same time. So then now we're just going to address a few misconceptions about evolution. There's three main ones. So first of all, it's not a theory. In science, theories are extensively tested. Evolution has been very extensively tested. And now we have, genetic evidence. We have all these pieces of data that we've recorded over time. So no, evolution is not just a theory. We know it's happened, and we know it's been happening and will continue to happen. Secondly, a big one, it's kind of hard to grasp, is that individuals evolve. No, they don't. Evolution, again, is a change in the allele frequency over time. It's a change in the gene pool, right? So, over generations. So, it's not just what happens in my lifetime, like I said, about going to the gym. Not a lifetime event, a generational event. And then lastly is that organisms evolve on purpose. So, due to natural selection, we kind of think that organisms evolve to be better suited to their environment or to better be able to accomplish something. No, that's not true. There are no goals in evolution. The only reason we evolve is because variation exists. That's a requirement for evolution, and that the environment or there's some form of selective pressure. Okay, so now talking about what natural selection actually is. So we have to understand the concept of fitness. We obviously have heard the term survival of the fittest. So fitness is based on most surviving and reproducing offspring. So the fittest organism will be able to produce the most offspring that survive, so they aren't just born and then immediately die, and go on to reproduce. So there may be a question that you come across, and you see how many offspring survive and how many offspring those offspring have, right? So their ability to reproduce. Sometimes it's not about the number of offspring you have directly, it's about how many more you have down the line. So again, just thinking about what makes the most offspring, what carries on those genes the furthest in time, that's the most fit. Okay, so survival of the fittest, that's where that comes from. Adaptations. So adaptations in an evolutionary sense refer to something that improves an organism's fitness in a specific environment. So how they happen is that natural selection, a pressure is going to select for a trait that acts on some variation in a population. And then lastly, putting this together, how does evolution by natural selection occur? Well, that happens when populations change over generations due to a fitness advantage. So natural selection works on some type of fitness advantage conferred by a trait. So a trait makes something more suitable to live in a certain environment, like the cold or the heat or in a desert, something like that. And that selective pressure makes a trait favored. Okay? So based on natural selection, we know that's happening. Well, because of that, we know that a pressure is going to cause traits that improve fitness in the environment. So throughout this population, as time goes on, populations will become more fit just because natural selection is favoring traits that make things more fit. And we actually know that because we're favoring only certain traits, genetic diversity will increase. So if you have traits that make you suitable to live in the tundra, in the cold, but you live in a desert, you're going to get weeded out by natural selection. So we're decreasing the diversity of those genes. And then environmental changes will change. Again, environmental changes is basically a way of saying change in selective pressures, because we know that natural selection is caused by some form of selective pressure, and so a change in the selective pressures or in the environment will change the average phenotype, which is the expression of traits. So hopefully we might have had an intro to bio class in high school or something before this. We know that the phenotype is the expression of traits. So that's the phenotype. And then just as a little side note, we have a genotype, which is the genetic makeup or the genetic composition of an organism. So the genotype is the genes that code for the phenotype. So the phenotype is what you actually see, it's what's expressed, and the genotype is what codes for that. Okay, a lot of arrows on the slide, sorry about that. So, moving on. What demonstrates evolution by natural selection? Okay, so again, looking at what we're looking for is some form of a selective pressure that causes selection for a trait that confers a fitness advantage. Okay, so we're looking for something being more fit than the rest of a population due to some form of pressure. So the first thing we're given is a cheetah. It can run faster, therefore it has a higher chance of capturing prey. However, looking at this right now, what does this say? A learned behavior. So its offspring learn the behavior, but is that a change in genetics? No, we have no change in genes. So this is not going to be evolution by natural selection. So looking at B, a population of lizards. So we grow larger due to an increase in food, where individuals change their diets within their lifetime, and these changes persist over subsequent generations. So looking at this, I'm thinking, no. And why is that? Again, because one, we see the key words within their lifetimes. So every time we hear that, it's kind of a red flag to like, no, that's not the answer. But also, we are growing larger, so we are changing, right? Is this change, right? If we're growing larger in our lifetime? No, because if you think about it, there's a statistic. Humans have grown a lot taller as time has gone on. That's not because of a change in our genetics or our DNA. It's a change caused by the amount of food we have available because of modern farming practices, because now we have better nutrition, which allows us to grow taller. So we're not evolving, we're just changing in our environment. So again, think about it. No change in allele frequencies, no change in our genes, so no, we are not evolving. Okay, next we're given one of the most classic depictions of evolution. We have dark moths and light moths, and the dark moths survive better against predation, and over time what happens is that the population shifts to become predominantly dark colored, okay? So yeah, we have a selective pressure, and then we have a result of that is that a certain set of traits is favored, so the genetics, the phenotype, shifts that way, so we know that our correct answer is going to be C. There we go. And then looking at it, D is not going to be right just because that's artificial selection. We see that there's some form an individual is selecting for a certain trait, not it happening naturally. I doubt there will be a question on artificial selection. Okay, so here are the impacts of natural selection. Okay, so the biggest one is that fitness will increase. So fitness increases. Why is that? Because the most fit traits are selected for. How adaptation happens is that traits are going to, the traits that confer a fitness advantage will be the ones that are selected for. And so what happens to the average phenotype is going to shift to increase fitness. We see this in these giraffes right here. More access to food means that over time we're going to grow a larger neck. And then genetic diversity, that is likely going to decrease because we're favoring a certain allele. Natural selection is not random. we're prioritizing one thing, and because we're leaning that way, we're going to decrease the prevalence of certain traits. For example, the trait that confers height among these giraffes, the trait that gives short height or not as long of a neck, that's going to not be favored for, while a large neck, tall height is favored for. So we're going to, over time, see an increase in that trait and a decrease in short height. So now we're given a practice question kind of based on what we just saw with the giraffes, where we see that we have a population of birds and we have a selective pressure applied by the availability of food, and that favors individuals with smaller beaks. And over time, that results in a decrease in the average beak size. And we're asked, how would natural selection explain this? So let's go ahead and look through our answer choices. And then when we look through this, I can immediately rule out two of them. And we can immediately go ahead, and we're looking at this, and we can go ahead and rule out C and D because we see this red flag right here. Individuals adjust beak size during their lifetime. Hopefully we know by now that every time we see it in their lifetime, it's not a form of evolution. And we can rule out D because it says researchers selectively breed. That's a form of artificial selection. So now we're between A and B. So looking at A, right, it says that birds with smaller beaks have higher fitness, which means that we have an adaptation and a decrease in beak size, and it maintains genetic diversity. So I can kind of get on board with this. Do we have a higher fitness because of the smaller beak? Yes, smaller beaks are favored because of the selective pressure of the food. So it's favored, which means that we're going to have a decrease in average beak size, right? Obviously, that makes sense because over generations that's going to happen, so we have more access to food. However, do we maintain genetic diversity? No. No, we don't. Because average beak size is favored. Not a large beak, a small beak. So genetic diversity will actually decrease. Okay? Let me actually write that out. No, it does not. Decreases. Okay? So now we're left to look at B, and it says birds with small beaks reproduce more, and that reduces average beak size and decreases genetic diversity. So yeah, we know that genetic diversity is going to decrease. So now let's really look at this one part. Birds with smaller beaks reproduce more. It's kind of hard to infer that from the question, right? Well, if you think about it, what does this have to do to be natural selection? Well, it's going to have to increase fitness, right? So if we're increasing fitness, what are we doing? We're reproducing more. So if we have more reproduction, that's going to mean an increase in fitness. We're going to have that fitness advantage. So we know it's going to be B because smaller beaks confer that fitness advantage. It says it, and that means that we're going to be able to reproduce more, and we're going to have these traits, the small beak trait, passed on more. So the correct answer is B. Okay, so looking at the types of selection that we can have, there are four main types, really three that you could think about graphically. So we have disruptive, stabilizing, and directional. So disruptive is going to prioritize either extreme phenotype. So for example, let's just say on one side we have the short beak, thinking back to those birds, and on the other side we have the large beak. And obviously in the middle we would have a medium-sized beak. So disruptive selection is going to prioritize either extreme, so either a large beak or a short beak. So some type of pressure that's like birds can't eat medium seeds, but they can eat large seeds and they can eat small seeds. So we're going to shift the average phenotype, the expression of those traits, both ways. So either towards a large beak or towards a short beak. So no medium beaks. Stabilizing is the opposite of that. We're going to shift towards the middle, towards away from the large beak, away from the small beak, towards the medium beak. So we're prioritizing the average phenotype. So both of these, disruptive selection, what's going to happen to genetic diversity? Well, we're moving from one average phenotype away to two extreme ones. So we're going to either maintain or increase genetic diversity. Now the average phenotype, that's going to go away, right? Because we're shifting away from that. Stabilizing selection, the opposite happens. We are shifting away from two extremes and we're going right to the average phenotype. So we're going to decrease genetic diversity because we're prioritizing one thing. However, the average phenotype, the middle, is going to increase, right? So just kind of think about these graphs, right? Disruptive selection, we have the two hills because we're moving that way. Stabilizing selection, we have a bell curve, we're moving right towards the middle. And then directional selection, hopefully you can get this one from the name, but we're shifting one way. So for example, in this directional selection, we're shifting towards the large beak. So we're going towards a single extreme. So obviously that's going to decrease genetic diversity because we're going to only one trait. And that's obviously going to shift the average phenotype towards that one thing. For example, on the past slide, the thing with the moths, where the dark moths are the favored for a trait, that's a form of directional selection because we are selecting for one trait only. And the last type that's kind of hard to think about graphically is balancing selection. So in this case, there is no shifts. It's literally, I like to think of this as, well, obviously all of them are preferred. So it's a rectangle in the form of our graph. So it would literally be a straight line like that, where all the average phenotypes are preferred, or a straight line. Anyways, the idea is here that neither trait, neither extreme nor the average trait confer any form of advantage. So now here is how evolution happens, and we have four main ways in addition to natural selection. So sometimes people call it the five fingers of evolution, where natural selection is one of them, and now we have the other four. Okay, so the first type is going to be mutations. So what is a mutation? Random changes in DNA. So this is just happening over time where DNA, what's happening in it is that hopefully you know, DNA is made up of four base pairs, A, T, C, and G, and sometimes over time, those just may randomly change. So this is going to have the smallest effect because it's just completely random. And most of the time, these mutations aren't good, right? Cancer is caused by a mutation. So most of the time, mutation is not going to be able to confer any form of favorable advantage. Next, we have gene drift and gene flow. So let's try, to me, they're pretty easy to get mixed up or to confuse. I know I made that mistake, so let's try and keep them separate. So genetic drift is when a random set of individuals pass on alleles within a population. So again, we are just changing the allele frequency in a population, caused by individuals in that population. So what happens is that it's mainly caused by either bottlenecks or the founder effect, where we have a limiting event happen, right? So these individuals, by complete random chance, are passing on their traits to the next generation. So again, random passing of traits by individuals in a population. Now, gene flow, the best way to think of this is like migration, like you're flowing down a river, because it's the movement of alleles between populations. So, for example, you completely join a new population, and now your traits are in the gene pool, and you start passing them down. That's a gene flow. And then lastly, we have non-random mating. Hopefully, we know what this one means by the title, by the name. Non-random mating happens when we have a preference for certain traits. This happens with peacocks, right? With the feathers. So mates are selected based on a certain trait, and that obviously creates a selective pressure towards those traits. So now here's some graphics of what those mechanisms look like. So non-random mating, right? We prefer a certain phenotype, and so obviously we're going to be selected for that. Genetic drift is where, again, due to random chance, these individuals end up passing on their traits. Then we have migration, aka gene flow, where one individual just carries those traits into a new population. And then lastly, we have natural selection. Hopefully we know that by now, where there is some form of selective pressure that's conferring a fitness advantage to certain traits. So for example, we have this dark colored and light colored bugs, and so, or beetles. In this case, the dark bugs are selected for, obviously. Okay, so now we're given this scenario where we have a population of wildflowers, and what happens? Oh no, a flood randomly kills a large number of them, and so it's going to reduce genetic diversity. And what explains this basically out of our different types of evolution? So what does the flood cause? Does the flood cause genetic drift? Does this happen by gene flow, non-random mating, or mutation? Okay, so immediately, let's go ahead and try and rule some of them out. Is this happening randomly? It's a flood, so there are no changes in our DNA. We're not mutating. We're not changing the base pairs. Non-random mating. Flowers are not able to sexually select like that. After the flood, they're not going to preferentially mate based on something like flower color. Flowers cannot arbitrarily do that, at least for the point of this class, and I mean, they're flowers. Okay, so now we're down to genetic drift and gene flow. So gene flow, migration. Are seeds blowing in after the flood? Are we introducing new alleles from a nearby population? No, there's a flood. Everything's been wiped out. We're not mutating. We're not selecting for flowers. We're not non-randomating, and we're not introducing new alleles. So when we look at this, a flood is a random event. It's a chance event. So when we change our allele frequencies, that's happening purely by chance alone. So the flood is causing genetic drift, which remember occurs randomly. And so it doesn't favor any specific traits, right? Blue flowers were not more likely to survive the flood than orange flowers. This was completely random, and it was selected for only by genetic drift. There we go. I forgot that we had a reminder slide. Okay, moving on. Now we're on to the second part, which is Hardy-Weinberg equilibrium and phylogenetic trees. Not my favorite part. Hardy-Weinberg is just simple algebra, so hopefully we can get onto this. So we have two parts of the Hardy-Weinberg equation. We have P and we have Q. So both of these are homozygotes. So P is homozygote dominant and Q is homozygote recessive. So that's where we work with our letters. So we have big A, and little a, and then obviously 2PQ. So PQ would obviously be big A, little a. So homo, and hetero. Homozygote dominant, homozygote recessive, and heterozygote. Okay, so now we have to understand that Hardy-Weinberg is basically a way of describing these allele frequencies within a population, and we know that p squared plus 2pq plus q squared is going to equal 1. Okay, that's just how these variables are arranged, and in order for Hardy-Weinberg to be applied, there are 5 assumptions that have to be made, and there is a question that's going to ask, can Hardy-Weinberg be applied to this population? You have to say yes or no and why. So for Hardy-Weinberg to be applied, for this math on the allele frequencies to be applied, there has to be no natural selection, no mutations, a large population, so we're resisting gene drift. So again, no natural selection, no mutations, no genetic drift, no gene flow, and no non-random mating, or just saying random mating. So basically, no. Just think about it like the 5 no's, okay? And then you have to know how to apply it, which means we also have to know the mathematical part of Hardy-Weinberg. P and Q, when you add them up, they will always equal 1. Same thing when you use the whole equation, p squared plus 2pq plus q squared is always going to equal 1. So no matter what, unless you've done your math wrong, they should equal 1. So whether these equal 1 does not tell you whether the population is evolving. That's just not how it works. So seeing how it works in practice, remember, P is our homozygote dominant, and Q is our homozygote recessive. So looking at this graphically, we have YY homozygote dominant, little y recessive, and YY our heterozygote. Okay, so now we have our frequencies. I'll go ahead and write P, PQ, and Q in here. So we have around 49%, 42%, and 9%. And what we're asked to do is calculate the total number of individuals. We're told that we have 500. So now we're looking at the frequency of big Y's, right? So we have 245 YY individuals, and we're looking at the frequency of Y's. So we're going to multiply this by 2. So we're going to get that 490 number because we're looking at the frequencies of the Y allele. And then we're going to take the big Y from this population of 210, we're going to move it down here. So we're told that we have 700 big Y alleles. How about little y? Well, we're going to take the 210 little y alleles from the heterozygote and the 45 from the homozygote, and we're going to get 300. So how are we going to find out P and Q? Well, we're going to take the 700 Y alleles out of the 1,000 total in the population. The 1,000 alleles, that's 700 plus 300. So we're going to take big Y over all the alleles, and we get 0.7 as P, and we get 300 little y's over 1,000 alleles total. That gives us 0.3 as Q. Now, one of the ways to analyze this is a Punnett square. Personally, I don't do this. I just set it up like algebra. But again, remember, we have P squared plus 2PQ plus 2 squared equals 1. So you could, if you want to do this algebraically, you could do 0.7 squared plus 2.7 times 0.3 plus 0.3 squared. You can do that, or you can do it with a Punnett square, where we have 0.7, 0.7, 0.3, 0.3. And we get the frequency of the homozygote dominant as 0.49, the frequency of the heterozygote as 0.21 and 0.21 respectively, and the frequency of the homozygote recessive as 0.9. Remember, we're just multiplying, so 0.7 times 0.7, 0.7 times 0.3 again, and then 0.3 times 0.3. I don't like using the Punnett square, but it shows us that the frequency of big Y is 49%, the frequency of the heterozygote is 42%, and the frequency of the homozygote is 9%. So that's how we would analyze Hardy-Weinberg. So again, we're asked to go through and do this for a population of 100 butterflies, but I already did it because Hardy-Weinberg sucks. But we have the allele big A, and it's dominant, and we have the allele little a, and we have 16 butterflies with white wings, and we're asked, we're told that the population is already in Hardy-Weinberg, so those 5 assumptions are true, and we're asked for what percent of the population is homozygote orange. So what do we have to do is we're looking for a homozygote, so we have to find P. Okay, so, or in this case, we have to find P squared. So we're told that we have 16 butterflies that are white wings, so little a, so we know that this is Q squared. So if Q squared is equal to 0.16, how do we find Q? We take the square root of that, we get 0.4. We know that P plus Q must equal 1, so P must equal 0.6. Let's square 0.6, and we get 0.36. So P is equal to 36%, and we can check this by plugging into P squared plus 2PQ plus Q squared and see if it equals 1, which it does. So 36% of the population is homozygous orange. Okay, so now we're given a chance to use Hardy-Weinberg to actually do some math with this. So let's go ahead, let's sigh, and let's get in the mindset to work with this. Of course. Okay, so we're told that we have 2 alleles, A1 and A2, and we have our frequencies 0.6 and 0.4, and we're told that the actual frequency that we're observing of the heterozygote, so the heterozygote we know is 2PQ, and we're told that it's 0.21, and we're asked whether or not the population is evolving or not. So basically what this is asking us to do is to do the Hardy-Weinberg math and see if the calculated values differs from the experimental or the observed values. So what we have to do is find 2PQ. So what we're given is P A1, which is equal to 0.6, and we're given Q, which is equal to 0.4. So now we need to find P squared and Q squared. So what is 0.6 squared? 0.36. And then what is 0.4 squared? That is going to be 0.16. Okay, so now that we have P squared and Q squared, we can go ahead and plug in for Hardy-Weinberg. So we have P squared plus 2PQ plus Q squared is equal to 1. So we have 0.36 plus 2PQ plus 0.16 is equal to 1. Can we solve for PQ by plugging in these values right here? Can we take these and plug in for 2PQ? Absolutely. Or we can just algebraically subtract 0.16 and 0.36 to solve for 2PQ. So 2PQ is equal to 0.48. That would be the same thing if you multiplied 0.6 and 0.4 by 2, where we get 0.48. So the expected heterozygote frequency is 48%. That's going to be A1, A2. Is that happening? Is the heterozygote 2PQ 0.48? No, it's 0.21. So yes, evolution is going to be occurring in this population. Why? Because we're differing from our calculated Hardy-Weinberg value. Yes, okay. Yes, we are, okay? We did the math and we looked at it and we have that difference, so we know that that's going to be evolution. So now we have the chance to do even more Hardy-Weinberg math if you really feel the need that you have practice. Okay, yeah. So we're told that we have 2 alleles in this case, G and little g, and they code for green or brown skin. And so out of our whole population, 240 frogs, we have 180 that are green and 60 that are brown. And we're told that this population should be in Hardy-Weinberg equilibrium, and we're asked to find 3 things. Big G and little g, the frequencies, P squared, 2PQ, and Q, and whether or not evolution is happening. Okay. where we have our green skin frogs and our brown skin frogs, and we're told that they can either be homozygous recessive or dominant or heterozygous. So let's start by finding P, Q, and then doing our Hardy-Weinberg math. Okay, so the only thing looking at this data that I can find right now is what Q is equal to. So we're told that our green skin frogs can either be homozygous or heterozygous, so we can't work with that right now. But the only brown frogs are homozygous recessive, so we can work with that. So we're not given a percent this time, so we have 60 homozygous frogs out of a total of 240, and when we do 60 divided by 240, we get so when we do 60 divided by 240, we get 0.25. So this Q value, this is actually for Q squared. So in order to find just Q, we can go ahead and take the square root of 0.25. And we know that the square root of 0.25 is going to be 0.5. And we know that P plus Q must equal 1. Therefore, P is also equal to 0.5. Okay, well, now we've done it, we can go ahead and figure out everything else. So P squared plus 2PQ plus Q squared must equal 1. So what's P squared? Well, 0.5 squared is 0.25 plus 2.5 times 0.5 plus 0.25 again must be equal to 1. And we know that 2 times 0.5 times 0.5 is equal to 0.5. So we have this definitely equals 1, so we know that our math is correct. And then, going back, looking into this, 0.25, that's our homozygote dominant, this is our heterozygote, and this is our homozygote recessive. So we should have 25% homozygote dominant, 25% homozygote recessive, and 50% heterozygous. Okay? And then look at this. Yes, P is equal to 0.5, Q is equal to 0.5, and then we have 25%, 50%, 25%. Is the population evolving? No, because, again, are we differing from this percent? So we have 240 frogs. I'll just work out an example of why we're not evolving real quick. So we have 240 frogs, 50% of them, or 25% of them, should be heterozygous recessive, so brown. So we multiply this by our percentage. And what do we get? We get 60 frogs. So we know that because what we're observing aligns with our math, no, the population is not evolving. Okay, now we're moving on to speciation. So what actually separates individuals by their traits into their own species? So there are 4 species concepts: biological, morphological, ecological, and phylogenetic. Okay? So biological, that's based on ability to produce offspring. So can 2 things mate and produce offspring that are fertile? Morphological, that's by physical. Ecological, that's based on niche. And phylogenetic, that's based on common ancestor. So for example, dogs are going to be the same species because dogs can breed with each other and make offspring that can also reproduce. Morphological, our example is owls because owls are going to be physically similar to each other. Ecological, that's based on niche. So niche. Please just say niche. Again, 2 types of bears, and then phylogenetic, common ancestor, Darwin's finches. They all have their own traits now, but they originated from one finch with a certain set of traits. Okay, so seeing how these can apply. So we're told that we have a population of birds that are isolated on islands. So like Darwin's finches, of course, they evolve their own traits. And we're trying to determine whether or not these separate traits should be considered a separate species. So which of the following applies the ecological species concept? And remember, the ecological species concept is talking about niche. So do these birds that have changed their traits have or do different things in the ecosystem? Okay, so A, island birds cannot interbreed with mainland birds, so they are separate species. So let's just say that's true. I mean, yeah, that would classify them as their own species, but it would be by the biological species concept, so the BSC. So they utilize their own niche, utilizing resources not by the mainland birds, so they're a separate species because they're fit in their own way into the ecosystem. So that is the definition of the ecological species concept, so the answer is B. But looking at C and D, well, C, they have distinct physical characteristics, such as beak shape. So what do we know? Well, that's going to be the morphological species concept because they're different physically. And then lastly, we're told that they form a unique branch in the evolutionary tree, so they're a separate species. That's based on a common ancestor or the divergence from one. This is the phylogenetic species concept. But we know that the correct answer is B because they're talking about a niche, and that's what the ecological species concept is. So there are two ways that speciation can arise. We have sympatric and allopatric. So the way I like to think about sympatric speciation as speciation due to time, where allopatric speciation is due to place. So a sympatric speciation, we have one population where as the course of time flows on, we have a second population. And again, just due to some type of difference occurring within the two populations, nothing's physically happening, but we end up with two separate species in the same area just due to time going on. So something causes an isolation event, and then boom, we end up with two species. Now, allopatric speciation, that's different because we're separated by a physical barrier or something that causes us to be separated by place. So we have our one original population, time goes on, something happens to separate our two populations, and then boom, we end up with two separate species because they physically cannot cross this barrier to reproduce, and then that's how we end up with two separate species. So sympatric happens in one area over time. Something causes isolation, but not physically. Allopatric, something physically causes reproductive isolation. Okay. So now we're talking about the two reproductive isolating mechanisms. So these are going to either be prezygotic or postzygotic. So pre and postzygotic is a fancy way of saying before or after the sperm and the egg of two species unite to form a zygote. Okay, so prezygotic obviously means before this happens. Okay, so prezygotic, I literally just took this chart because it's just so good. Prezygotic, there are four types. So we have temporal, which is due to time, ecological, which is due to niche, behavior, which is obviously due to behavior, and the mechanical, which is due to physical differences. Okay, so looking at this temporal, the example is that these frogs breed in the summer, these frogs breed in the winter. They cannot breed just due to that time difference. So it's prezygotic because they don't even get the chance. Ecological, that's due to the niche. Lions and tigers, can they interbreed? Sure, but do they do different things in the environment? Yeah, so are they going to actually breed? No. So they're separated reproductively by that difference in niche. Behavior, these birds, they're not going to mate with each other because they have different mating calls. Could they? Probably, but they're not going to because of that difference, so they're not going to breed. Sperm and egg don't unite. That's going to be before, and that's the behavioral type. And lastly is mechanical. So can these two breeds of dogs mate? No, because of a difference in physical size. So that's going to all be before sperm and egg unite. And then lastly, we have postzygotic. So that's what happens to the actual offspring that they have. So first we have inviability of the hybrids. So when we say hybrid, we mean that basically two species come together and they form this mix, a hybrid. So when we have an inviable hybrid, sure, we go off and we have this offspring, but can it grow on? No, it cannot. So basically, we've stopped any further reproduction right there because we haven't grown into a full frog. Inviability, we're not growing to adulthood. Now, infertility, here's what happens. Yeah, we grow into adulthood. Yes, we become a full-on mule or a donkey or whatever this is. But can we go on and then reproduce? No. So hybrid infertility means that the hybrids are sterile, and hybrid inviability means that we just won't grow to adulthood. So both of these mean that hybrids are at a fitness disadvantage. So species are going to be reproductively isolated from producing hybrids because we get a fitness disadvantage, and no one wants that. And then lastly, the last type is a hybrid breakdown. Doesn't really happen, but it just means that yes, we get perfect offspring, so we think that nothing's wrong, but then those offspring have offspring and they don't grow. So hybrid breakdown rarely happens. Mainly it's inviability or infertility. Basically, those are postzygotic and prezygotic isolating mechanisms that lead to the process of speciation. Okay, so how do our types of evolution lead to speciation? So what happens? Gene flow makes things more similar, so homogenizes, which decreases speciation, where the rest, genetic drift, selection, and mutation, unhomogenize a population, and they create divergence. And then non-random mating, this is the big way that reproductive isolation can occur. That's what we just learned lastly. So reproductive isolation, pre-post-zygotic, that happens because of non-random mating. So pre and post-zygotic barriers, that's before or after sperm and egg unite. So for example, this bullet point is wrong. Behavioral differences, an example of prezygotic, sperm and egg being incompatible. Is that prezygotic or postzygotic? Actually, no, it's not wrong. Because if sperm and egg are incompatible, that is an example of a mechanical difference. Because we physically cannot form that zygote, so it's prezygotic. However, if we go ahead and we don't grow, or we're spontaneously aborted, so we can't grow as a hybrid, that's going to be a postzygotic isolation mechanism. And then again, look at this, a hybrid is caused by interbreeding of these two species. And then what happens is that hybrids are going to be less fit, right? Inviable or infertile, and that causes a fitness disadvantage. So we do not want to make hybrids, and that causes reproductive isolation. So now we're given this question where we're asked how one of these mechanisms can lead, in speciation, can lead to reduced hybrid fitness, which leads to prezygotic isolation. So before sperm and before. So now we're asked how these evolutionary mechanisms in the speciation result in reduced hybrid fitness, which leads to prezygotic isolation. So again, before sperm and egg unite. And so basically, what these scenarios are, let's look at the first one. So we have genetic drift causing flower color differences that prevent cross-pollination, so some form of mechanical prezygotic isolation that results in speciation despite, here's our red flag, despite high hybrid viability. So does this demonstrate how reduced hybrid fitness leads to this? No, so it's not A. Then we have B. Gene flow between two bird populations reduces hybrid fitness, but there are no prezygotic barriers. So now we have not reduced hybrid fitness levels, and we have no prezygotic barriers. So it's not going to be B. Now we have C, where we have two frog populations. They evolve distinct mating goals, so a behavioral difference. And then when they can actually mate, when they can interbreed, we have low hybrid survival. So what is that called? That's called hybrid inviability, and we favor prezygotic isolation. Well, I think we found our answer here because we have a prezygotic isolating mechanism, the behavior, and we have hybrid inviability, which leads us to have a form of prezygotic isolation. Look at the last one, we have mutation, where non-random mating leads to reduced hybrid fitness, but we have continued interbreeding. So obviously that would not be it because we're still doing it. So the correct answer is C. Okay. Now we're talking about phylogenetic trees, which is a way of showing the relation between species as they come or as they evolve off of a common ancestor. So here are the parts of a phylogenetic tree, right? So the roots are at the very end, right? So so now we're asked, so now we're talking about phylogenetic trees, which are a way of showing the relation of species as they diverge and evolve off of a common ancestor. So just looking at how to read one of them. So the first thing we're looking at is the root of a phylogenetic tree, and that's going to be the origin of a phylogenetic tree. So right here at the very start of the phylogenetic tree is our root. Then we're asked about the nodes, which is where things branch off of. So branching point. So again, looking here, here's a node, here's a node, here's a node. I don't know why the node here is not labeled, but yes, it is a node. So things are branching off of each one. Then our branches are the actual things coming off of the nodes. All right, so this is a branch, this is a branch, all these things are branches, and then the tips. I'm not going to label those because hopefully we can look at the end of the phylogenetic tree and identify what a tip is. Okay, the next thing that we're asked to identify is a monophyletic group, including nested monophyletic groups. So a monophyletic group, and for the rest of this, because that's a long word to say, I will be calling a clade. They are the same thing, but they are a group of species and a common ancestor. So for example, looking at the clade here, we have species A and species B. So if we go and we draw a box around them, it encompasses the node, it encompasses a common ancestor, so it's a monophyletic group. Here we have C, D, and E, so we draw a box around it, we encompass a node and all our common ancestors. So we are coming down from here, and that encompasses C, D, and E. So what about D and E? D and E, yes. But what about B, C, D, and E? Are we a clade? No, because we don't include the common ancestor of A. Hopefully, identifying clades is kind of like intuitive to you. So for example, are B and C monophyletic here? No, because we're not including the common ancestor back here. Are A, B, C, and D? Yes, because we go all the way back to here. Are A and B? Yes, we go all the way back to here. Are A, B, C, D, E, and F? Yes, because we go all the way back to here. What about C, D, E? No. So it's just about whether or not we're encompassing everything, going all the way back on a phylogenetic tree to encompass the species plus a common ancestor. So when we're making a phylogenetic tree, at the end, at all of our tips, we have a clade, basically. So this is actually a phylogenetic tree that we use in BIOS 1108, so bioprinciples 2. So again, at the end, we label or we put our monophyletic groups. So these in their own could be their own monophyletic or their phylogenetic tree, but we put them at the end at the tips. So it's just an interesting way to think about it. Again, we're modeling the relationships. So for example, fungi and animals, yeah, that's a clade together because they share this common ancestor. Protists, fungi, animals, and plants, they are also a clade because they share a common ancestor. They became eukaryotes. Archaea and bacteria, no, because look at it's not encompassing everything. Now, bacteria, eukaryotes, and archaea, yes, because they all share this common ancestor at the root. So now we're asked to identify synapomorphies that identify a monophyletic group. So synapomorphy is just these traits that have evolved together. So for example, a gizzard. What is our clade going to be for the trait of a gizzard? Well, it's going to be the bald eagle and the alligator because they both have those. What about lungs? Well, now we have the antelope, the bald eagle, and the alligator. What about fur? Is it going to be the bald eagle and the antelope? No, because only the antelope has fur. And what about jaws? Everything but whatever the lamprey is. I don't know what that is. I don't want to know. So again, just looking at what traits do all these things share. So again, going through that and going through with this process, what are some of these common traits? Hair, we see that in these primates. What about an amniotic egg from the primates to dinosaurs and birds? What about four limbs from amphibians to dinosaurs and birds? So we just go down and we see everything that shares this trait as it evolves. Yeah, so there we go. Hair, yeah. So now, we're trying to evaluate the most common ancestor to evaluate the relatedness of species. So evaluating relatedness, how far away have they branched off? So for example, crocodiles are more related to dinosaurs and birds than rodents and rabbits because we had all these branching points that it takes to get up to dinosaurs and birds. I know I'm annotating a lot here, but just think about it like that, right? Like sharks are really far from being related to dinosaurs and birds. Primates are closer related. Amphibians are closer to sharks. And just seeing how far each thing is off of this branching point. So, for example, sharks only have vertebrae, so that's 1. Bony skeleton, 2. That's how we get to the ray fin fish. What about amphibians? We are 3 away, and just we go further and further. That's how we identify the relatedness of species by how many traits or how far they away are they on the phylogenetic tree. Common misconception, I don't know if another slide covers this, but things are not more evolved than each other. A phylogenetic tree represents time, so all the way up until now, this arrow is now. So all of these things are not more evolved than one another. They are all equally evolved. So that's a common misconception. These are all equally evolved. The number of traits doesn't matter. They have all made it to now, to current time, so they are all equally evolved in time. And then how do we make phylogenetic trees? It can be morphological, so physically, or it can be genetically based on like mitochondrial DNA, ribosomal RNA, or any genes. And what they're used is to identify similarity due to a common ancestor. And this is important. This last bullet, it's based uh, on what's most parsimonious. Okay, so on what is what's simplest, basically. So for example, when we're looking at a trait, what requires the least amount of evolution is likely to be the most likely explanation of things. So for example, saying that hair evolved separately in rabbits and primates is less parsimonious than saying that they both evolved hair and grew it. That's how parsimony works. And then lastly, we're on to my favorite part of this unit, which is on Earth history and how life actually originated. Look at this meme here. DNA is superior to RNA. Okay, I don't know why. There are some scientists that think not. No. DNA, superior form of genetic information. And all life uses it. So, you know. So, looking at Earth history, we're looking at the layers of the Earth. That's something called stratigraphy. So, you might be asked to look at a set of layers, and identify which is the oldest, which is the youngest, yada yada yada. So luckily, things layer oldest to youngest, right? So the bottom layer is obviously going to be the oldest, no matter what. Even if something has been like cut into it, we're still going oldest to youngest. The only exception is if something is cutting across, right? So the youngest thing is likely to be what's cutting across. So we have 1, 2, 3 layers, and then we have this cut, which is the youngest, only to be buried by the top layer. So applying that right now, what is the oldest and what is the youngest? So the youngest, that's going to be the top layer. The oldest, that's going to be the lowest layer. So let's literally go down and identify those right here. Top layer, lower layer. And then if we want to, just to show the cross cutting, so this is oldest, so I'll put a 1, and then 2, and then a 3 on this layer, and then 4 on this blue layer. And then we have this cut here, that's 5, and then 6, we're layering on top, 7, 8, 9, 10. So this is our youngest layer here, the 10, this is our oldest, and then the cross cutting is younger than everything until the layer on top of it. And there we go, youngest and oldest. Now for radiometric dating, that's how we identify how old rocks and fossils are based on elements that decay over time. For example, carbon-14 and carbon-12. By comparing the ratio of those two, we can see how old something is. Luckily, we're not really asked to do too much with that in this class specifically. We just have to know how things decay. So the big concept is the half-life, right? So as we undergo one half-life, we go from, let's say we have 100% carbon-14. We undergo one half-life. We have 50% carbon-14. Another half-life. We have 25% carbon-14. So as time goes on, we can compare the ratio of carbon-14 to carbon-12. Because after one half-life, we have 50-50 carbon-14 to carbon-12. And then we have 25% carbon-14, but 75% carbon-12. So that's just comparing those ratios. As that number of half-life goes on. And then to figure out the age, we take the number of half-lives that have elapsed multiplied by what the actual half-life in years of the isotope is. So for example, in this question, we're told that we have carbon-14, and it has a half-life of 6,000 years, just to make things easy on us. And then a fossil that is 1/16th the age, that's 1/16th the normal proportion of carbon-12, should be how many years old? So again, looking at our ratios here, 1/2, 1/4, 1/8, 1/16 as the number of half-lives increases. So what do? Well, we take the number of half-lives, which in this case is 4, multiplied by the actual half-life itself, 5,700 or 6,000 in this case, and we get that number. So probably 24,000, but in this case, if we use 5,700, 22,800 years. So that's the most we'll have to work with these. It should not be that bad at all. So again, here's our practice question. In this case, we have 40 potassium and 40 argon. Potassium decays into argon, and we have a half-life of 1.25 billion years. So if we have no argon initially present, so at this point, initially we have 100% potassium 40. At what point will we have equal amounts, so 50-50 potassium 40 and 40 argon? So literally one half-life, and we're going to have 50% potassium 40 and 50% argon 40. Okay? And so identifying this right here, we can say that after 1.25 billion years, one half-life is when we'll have a 50-50 mix. Yep. And that's all we'll have to do. Now we're just talking about biological events as they happen. So what happens is that initially, basically, the big biological innovation here is the ability to perform photosynthesis. That's the biggest thing. I'm going to write that out. We're going to talk about this in the next slide. But why is photosynthesis important? Because it produces oxygen. Remember, early things did not need oxygen, so looking at this diagram, atmospheric oxygen, it used to be at 0%, but then, boom, it increases like crazy, and we get the Cambrian explosion right here, where a bunch of eukaryotes grow, and now we have modern life right here. So now we're asked about an adaptive radiation. So an adaptive radiation is just this massive change in the traits and blossoming of a new species, normally caused by a mass extinction. So an adaptive radiation happens, and this is crucial, when a new niche becomes available. So normally after mass extinction. So for example, we had the event that produced a lot of oxygen, a new niche evolved for organisms that could use oxygen. So we had an adaptive radiation of all these organisms that can now use oxygen, and that's where we get our Cambrian explosion. So again, here is an example, a practice question on how something can cause an adaptive radiation. So in this case, looking at it, what causes the most niches to be available? So looking at these immediately, we see a stable environment. No, because there's not a lot of niches being freed up. Minimal ecological changes. A single species evolves a key innovation such as photosynthesis in an already saturated ecosystem. So, right here, this is going to cause a massive mass extinction here, but we're not diversifying. So have we caused mass extinction? Yes. Have we opened up new niches? Probably, but have we radiated out? Have we blossomed in the number of organisms? No. Climate stability in millions of years prevents mass extinction and adaptive radiation? No. So we need those new niches to open up. That's going to happen in our mass extinction event. So A is our correct answer. Okay, so here are the main events that happened. We have early Earth. So we have, during early Earth, we have low oxygen. So most organisms don't even use oxygen. Then we have the evolution of photosynthesis. And that gives us oxygen. So now we're gradually increasing oxygen levels in the environment over billions and billions of years, and suddenly, boom, Cambrian explosion caused by the availability of oxygen that allows higher level life to grow. We can see that on this chart here. So we evolve it, and we start producing oxygen, and then billions and billions or millions of years, we have the Cambrian explosion where things start being able to use oxygen. It's an adaptive radiation. Okay, now, how did life actually originate on Earth? So we have this Halting hypothesis with how we made the primordial soup for which all things on Earth originated from. So what happened is that, remember, early Earth was all just water. So we have this boiling flask. It's filled with water and a bunch of other, stuff that would be in the atmosphere: methane, ammonia, water vapor, hydrogen gas, all this stuff just floating around in the water. We have a spark, which in this case simulates lightning. So lightning, boom, it hits all this stuff in the atmosphere. It turns it into all these different elements. It goes down and it enters the ocean. And then suddenly we have all these organic molecules from that spark of lightning that can grow and form into the proteins and the amino acids and the components of life. So basically, for life to originate on Earth, we need a form of hereditary information, right? Because we need our genes. We need to be able to use energy. We need to be able to grow, and we need to be composed of organic molecules. That's the foundations of organic chemistry, my type of chemistry. So an organic molecule contains carbon. That's the definition in biology. Typically, we also have hydrogen, oxygen, nitrogen, phosphorus, and sulfur. That's the composition of DNA, proteins, and fats. So we need those in order to be alive, basically. So what happens is that we ran this experiment. We saw that organic molecules could be produced from inorganic molecules that were just sitting in the water, sitting in our atmosphere. And then now, basically, it happened in 4 steps. So first, we form those organic molecules from the lightning. Then those organic molecules stick together to form polymers, and those polymers begin to have functions other than just existing and being a structural molecule. So proteins and RNA form, and both have some function metabolically or as enzymes, which speed up reactions. So RNA can be catalytic, which means it speeds up a reaction. RNA also encodes hereditary information, which means that now we get protocells. Protocells are not alive, but basically because we formed fats, fats do not like water. So what they do is they're going to clump up, and when they clump up, they surround a bunch of organic molecules. So they're protocells, so they're not alive, but they are the foundations of what modern cells are like because they have a membrane. So we kind of identified now today that all life originated from a common ancestor because we have a DNA-based genetic code, a lipid membrane, and we have proteins. Everything on life, bacteria, archaea, and eukarya, across the 3 domains, all have these commonalities shared between them. So now we're asked to summarize basically what the requirements for the origin of life are and how they happened. So remember, we started with these organic molecules that were synthesized abiotically, so without any organism doing it, just by chance, by lightning. Those organic molecules formed polymers, and then they became protocells that could replicate, and then now today we have universal life. Okay? So let's see. Looking at these options, life did not originate in hydrothermal vents. No, no, no. It originated in, I'm going to answer this one just to be able to talk through it. Life originated in a reducing atmosphere. So the answer is C. I know B might have looked tempting, but the answer is C because this atmosphere that the hypothesis was conducted in is reducing, which means that there are free electrons to be gained. You'll learn about that in our biochemistry unit. But basically, now we have LUCA, the last universal common ancestor, and identical cell structures, meaning our membrane, our DNA, the use of proteins, all those things that show that. So now we're asked to take all those requirements and put them together. I'm just going to talk through this real quick. So the origins of life, right? We started with the abiotic synthesis of organic molecules. It just happened by a spark of lightning. There were no organisms to do it. So those were synthesized completely by chance. And then those organic molecules started to stick together to form polymers. Those polymers, such as RNA, began to replicate, right? Both catalytic and genetic functions. And then now, today, after they started replicating over and over and over, and life changes over millions and billions of years, now we have universal evidence, such as the use of ATP and the presence of ribosomes, that mean that we have a common ancestor. So the correct answer is B. So now, looking at LUCA, LUCA stands for Last Universal Common Ancestor. So again, everything in bacteria, archaea, and eukarya, the 3 domains of life, originated from LUCA. So LUCA shares all these traits, which is the use of a DNA-based genetic code, a lipid bilayer, so lipid membrane, and the use of proteins. So we all have these in common with LUCA. So there had to be some form of early cell that everything diverged off of. So again, looking at what was actually inside LUCA, we have this membrane, and then we have our genetic code in the form of DNA, but then we also have RNA that we use today. We have enzymes, and we're able to do all these things with the organic molecules in the water and in the atmosphere. And I think LUCA is really cool at how life originated. So now we're asked, what's present in Luca? And hopefully we know this now, right? Let's look at these. A membrane-bound nucleus. Do Luca's the universal ancestor to all three domains of life. Do prokaryote, well, do bacteria and archaea have nuclei? No, so it's not this. Can bacteria, archaea, and eukarya all perform photosynthesis? No. Do we have a fully developed mitochondrial system? No. So the answer is the use of DNA-based genetic code. We see this right here. All three only have DNA, membranes, and proteins in common, so the correct answer is DNA. So now we're just kind of talking about how life originated, and this is called the RNA world hypothesis. So why then? Everything today uses DNA, but then why was the first genetic material RNA? So basically, it's because of this dual function, where RNA both can replicate and catalyze their own reactions. So basically, DNA, when we replicate DNA today, we have to have enzymes, and we have to have something called a primer. You'll learn about this again in the biochemistry unit. But RNA does not need any extra enzymes because it itself is an enzyme. And because it's catalytic, it means it can not only store genetic information, but it can replicate itself. So that just gave it this, again, a selective advantage all the way back then that made it more favorable. So talking about prebiotic, we can't really talk about evolution because RNA is not alive. But if we're thinking about this with natural selection, if we have a bunch of different RNA strands, because again, all by random chance, all just existing, floating around, the most catalytically efficient RNA is going to win out over the rest, right? So there's also kind of like a universal strand of RNA that beat out all the rest because it could reproduce the fastest, which means we ended up with the most strands of it. So if it could reproduce itself a bunch of times and with the least errors, so without changing its base pairs a bunch, that would be the strand that's selected for based on just, the fact that that's how natural selection works. It's the most favored and in a way has fitness because it's reproducing the most. So again, that's how that's our practice question where we're asked how natural selection can apply to a chemical system. So we can't really talk about evolution because it's not happening in a live system. But we can again see that when we have these molecules that can replicate, there is some form of selection going on. And in this case, we have self-replicating molecules that resist degradation and that replicate more frequently. So we have two forms of selection that give it an advantage right here. So our correct answer is C, right? So without any variation, no selection, degrade at the same rate. There's no benefits to that. And then randomness. So again, none of these are the correct answer. C is the correct answer because we have some kind of selective advantage. And there we go. That's the end of Unit 1. That takes us all the way from what life is to how life originated with RNA and Darwin's finches. So yeah, that's all of life and evolution, and good luck with your studies.